Hello students, welcome to Microbial Cell Culture and its application lecture 5. Today we are going to study about the downstream processing. So, what we have done so far is we have seen why biotechnology is needed, then we have seen where the what kind of cell culture techniques are there, what kind of equipments are there, what kind of nutrients cell requires. We have seen in equipments incubator shakers, fermenters, bioreactors. Then we have also studied the stirred tank bioreactor. After that, we have seen what kind of what kind of reactor settings that you can work with. It can be fed batch reactor, it can be continuous reactor. After that, we had looked at the principles of biomass measurements, cell number using identifying the doubling time using cell numbers, and also identifying doubling time with the biomass. We have also seen how actually to measure biomass using spectrophotometer as well as colony forming units CFU, or just by measurement of dry weight. We have also seen what are the challenges in scale up and what is the requirement of pilot, where it comes into the picture. Okay. So, today we will see that now we have done all this, now we have produced our product. Now comes that we take the product out, purify it and put it in a marketable form. So, that is called as isolation of the microbial product. It is also called as typically it is called as downstream processing or biosuppression. The reason to do it is to get the desired product in marketable form, because you cannot sell a broth to the customers. You need to have it in a packaged, uh, well packaged, as well as which makes it attractive for the customers to buy, as well as easier to transport. So, we need to get the product in the marketable form, as well as we need to purify the product. So, we cannot sell a product which is 10% pure. We need to purify the product to 99.99% if it is in therapeutics, around 98-99% if it is in diagnostic, and it would be above 90-92% if it is an industrial product. And depending on its application, the purity can vary, but the contaminants cannot be such which interferes on the post use of the product. So, we need to make sure that we remove those contaminants. So, this all forms the part of downstream processing. Why we want to understand it? There are two reasons for that. One is, as I said, we need to increase the purity, but at the same time also increase the concentration. So, if you increase the purity, the product concentration becomes more and more, and this is the most dominant one. Okay. And another thing is that overall of the bioproduct formation, 60% of the total product cost is by downstream processing. So, to recover the product, remove it, remove from the cell, remove the contaminants, remove the very small amount of contaminants which are there, which are not in higher quantity, but if they are present, then they impact the post use. For instance, viruses can be only 1 or 2% or 3% of the contaminant in the bioproduct, but this 3% in therapeutics is absolutely no go. So, you need to remove this 3% to bring it down to 0.001% or something like that, which will be an acceptable value. So, we need to remove them, and then essentially also we cannot sell a liquid. In some cases yes, but many times you need to have a formulation, okay, and for that we need downstream processing. Okay. So, these are the typical steps in the downstream processing, or typical thought process in the downstream processing. So, there is removal of insolubles. So, what is the insoluble over here? The first insoluble over here is biomass. We need to remove biomass. Okay. After that, we do a isolation of the product. Okay. Isolation of the product means we want to take out the product from the major contaminant, but at the same time you will see that we are also taking out our contaminants with the product. Okay. Typically solvent extraction, adsorption, these are two processes, two unit operations which take place in the isolation of product. Then we have purification and up concentration. liquid chromatography uh, is essentially used in purification. Okay. We can also use to a certain extent sometimes precipitation, but liquid chromatography is go to method for purification. However, liquid chromatography always leads to dilution, it never up concentrate the product. So, if you have taken the product, then if you do an solid phase extraction unit operation, then you will have an up concentration, and then you do polishing and packing. Polishing step is typically crystallization. Crystallization is a process where basically you make crystals. For instance, if you have a salt solution, a dissolved salt, okay, NaCl, our common salt that we add in our food. If you take that common salt in the water, you mix it well, it is soluble, and then you allow water to evaporate. You will see the formation of white powder. That actually is crystallization, but it is actually not white powder, it is white crystals. The difference between powder and crystal is that in crystals you can define crystals with uh, with coordinates, and it is a repeatable units. So, if you give A, B, C an angle alpha, beta, gamma and an angle, then each, if you keep on joining them, you form a crystal, while powder or amorphous you cannot really define them, they have no particular shape. Okay, they are completely random. So, these are the 4 steps involved: removal of insolubles, isolation of products, purification and up concentration, and polishing and packing. We will go into details of each of the steps when we have a look at it in the next couple of slides. What is the fundamental of downstream processing? So, if I have to sum it up in couple of words or in just 1 or 2 line, the fundamental is exploiting the differences in the property of the product and impurities. This is the fundamental of downstream processing. Essentially what it means is that we want to remove product from the impurities, or we want to separate product from the impurities by exploiting the difference in their properties. Now, what property that we are talking about? It can be properties based on affinity. So, for instance, a product is hydrophobic uh, while our impurity is hydrophilic, or the other way around, that our product is hydrophilic while our impurity is hydrophobic. It can be that our product has a tendency to form covalent bond while impurity does not. It can have strong electrostatic interaction. It is our product is keen to form complexation with metals while the impurities does not. So, this kind of properties that can be used to exploit it. This properties can be exploited for downstream processing. The other property can be solubility. Some products are very soluble in water while absolutely insoluble in other solvent. Okay. While our impurity is soluble in the solvent, so we can actually move the impurity from the water to the solvent and get the product in the water, or the other way around, which is more common, where our product is more soluble in the solvent while the impurities are not soluble in the solvent. A typical example of this would be antibiotic. Antibiotics like penicillin is very soluble in amyl acetate, while the product is not, while our impurities are not soluble in amyl acetate. So, basically you move the impurities, or you move the product, not impurities, you move the product from our aqueous phase to amyl acetate, the solvent phase. Okay. Then it can be stability, temperature or pH. So, at certain pH values the product is soluble, while at certain pH values the product is insoluble. For example, glutamic acid, if you decrease the pH to 3.2, it will precipitate, and then you just collect the precipitate and remove all the rest of the impurities, but you have to make sure that the impurities are not also precipitating at that temperature, at that pH. Also temperature plays a role. For example, desferoxamine E, it is a siderophore molecule. This actually precipitate at lower temperature while the impurities will not. So, all of these things actually comes into the play for separating the product and the impurities. Okay. How basically we can identify for the chemical, how basically we can identify what kind of properties the molecule has. Okay. For that we have host of tools such as HPLC, that is high pressure liquid chromatography, high performance liquid chromatography. So, it is not pressure, it is high performance liquid chromatography. We have IC, it is ion chromatography. We have ICP-MS, that is typically used to measure metals. Okay. It can actually only measure metals, but metals at microgram per liter or even nanogram per liter concentrations. Okay. Then we have FTIR, this is infrared spectroscopy. Basically, it can give you differences between an carboxyl bond, peptide bond or a hydroxyl groups, and so on and so forth. Similarly, NMR, this is nuclear magnetic resonance. This also gives an information on carbon and bonds, which kind of bonds are there, what kind of surrounding atoms are there, and what bond is vibrating. Okay. Similarly, Raman spectroscopy does the same thing. UV visible spectroscopy, you have spectro photometer, you have seen in our last class how it can be used. Then you have a SEC, that is size exclusion chromatography. So, it separates the products impurities based on the sites. Then you have LCMS, that is liquid chromatography mass spectrometry. So, it is much even more sensitive than your high performance liquid chromatography. And then you have LCMS-MS, which is even much more sensitive than LCMS, and you can do much more things with LCMS-MS than compared to LCMS. Okay. So, this all this information, this analytical tool is just for your knowledge, so that you know that there are multiple of analytical tools available to assess the properties of the biomolecules or your products, impurities and everything. Okay. And in any case, you would know that what kind of impurities you have, what kind of products you have, and you would know the structure of the product. You are not going to go completely blindfolded. You would have an idea, you would have some guesswork, you would have already investigated this while producing product, also while doing the batch culture in the shake flask study. So, you would know what kind of impurities you can expect, what is the product properties that you can expect, and after that you can use these analytical tools to give more quantitative values as well as qualitative value. So, quantitative means you can talk about concentrations, milligram per liter, microgram per liter, grams per liter, okay, or moles per liter. Qualitative means that you can say, okay, carboxyl bonds are present, hydroxyl bonds are present, but you do not know in how much value, in what ratios. Okay. So, that would mean that is a qualitative measurement, while quantitative is we can give an absolute value. Okay. So, the takeaway message from this slide is this: you need to know what is the properties of your product, and also at the same time what is the property of your contaminants. You can see and evaluate what is the difference between these two property is, and basically then use these properties to essentially separate the compounds. Okay. So, that is one thing. Second thing is that how do you know these properties? There are several analytical tools that are present. So, high performance liquid chromatography, ion chromatography, ICP-MS, infrared spectroscopy, size exclusion chromatography, liquid chromatography MS, mass spectrometry, and so on and so forth. So, there are multiple of analytical tools are available that can help you do it, and some of this tool actually can be used to separate the product as well. So, for instance, liquid chromatography is very much used for separating the product. Ion chromatography is very much used to separate the metals or take out the metals from them. Okay. So, size exclusion chromatography the same. So, this some of these analytical tools are also there for separation or for doing the getting the desired product in desired purity and quality and quantity and so on and so forth. Okay. So, these are the fundamentals of the downstream processing. Let us have couple of examples. Okay. So, we want to exploit the difference in the property of the product and impurities. So, let us say you have a product glutamic acid which is on your left hand side of your screen, okay, and then furfural on the right hand side of the screen. Okay. how would you go about separating them? So, I would like you to focus on the structure. Just have a look at the structure first. Is it easy to assume that glutamic acid looks like more hydrophilic while furfural looks like more hydrophobic? Right? Because of the ring structure, while because of the amine group in the glutamic acid, so NH2 group and OH group, COH group, it looks like more hydrophilic molecule. So, one way of the separation would be exploiting the hydrophobic interaction. Okay. So, let me just explain this thing. Okay. You have COOH, okay, and NH2, okay. This is your peptide bond, and then basically you would have side chain, and at the end you have COO minus or COOH, whatever it is. Okay. This is your glutamic acid. Okay. How does your furfural looks like? Furfural looks like a pentone type structure. Okay. Okay. This is how your furfural would look like. Okay. So, this is your furfural. Okay. What you can do with it? See, because of the charges I can see that this is more hydrophilic, this is more hydrophobic. Okay. So, I will write it here again, this is more hydrophobic. Okay. So, you can exploit if you have a beads. Okay. So, beads like this, okay, and they have some carbon chains, let us say C18 chains. Okay. So, what does this C18 chains done? It promotes hydrophobic interaction. Okay. When it promotes hydrophobic interaction, then basically this bead would attract furfural while repulse your glutamic acid. Okay. So, what can we do? What can we do is the following. So, what can we do is the following. We have beads, we have carbon chains C18, okay. We add furfural here, we add glutamic acid here, okay. It both of them added, then what happens is that you have a bead, and your furfural remains here while your glutamic acid pass through. Okay. So, now what you have is bead with furfural attached. Okay. So, how that will look like is like this. Let us say our red is furfural molecule. Okay. So, this is our furfural molecule, while our, if you have liquid going in, so short form furfural, okay, furfural, and another short form of glutamic acid is glue passing through, furfural remains here and glutamic acid will come here. So, why that happens is because furfural is an hydrophobic molecule and the bead is also hydrophobic molecule. So, they interact with each other and this is basically a hydrophobic interaction, and hence furfural is attached to the column. So, this is interaction taking place, while glutamic acid cannot interact and thus comes out. Okay. And then how do you recover furfural? Now, you have beads again, you add solvent, which is more hydrophobic, you add solvent which is more hydrophobic, you add a solvent which is more hydrophobic than furfural, and what will happen is you have bead again, okay, you have carbon chains and your solvent would come here, while your furfural would get separated, and this is your furfural. What I have explained you here is actually an adsorption process system. Okay. So, this is how basically you can exploit the difference in the properties to actually separate glutamic acid and furfural. Okay. Let us look at another example. Now, you want to separate penicillin from biomass and glucose. Okay. How can you do it? So, let us first look at penicillin separation from biomass. So, what is the property of penicillin? That can be used. Okay. Let us list down its properties. Is it soluble in water? Of course, it is also soluble in amyl acetate, but it is also soluble in water, and it is more soluble in amyl acetate as compared to what it is in water at certain pH values. Okay. So, penicillin is soluble in water. What about biomass? Is it soluble in water? Or it is insoluble in water? It is insoluble in water, it cannot be solved, it cannot be soluble in water. Okay. So, if it is insoluble in water, what techniques we can use? Can we exploit this? Can we exploit the difference in this property of penicillin and biomass, where a penicillin is soluble in water while biomass is not soluble in water? What can we use to separate them? So, there are many ways actually to separate them. One is, of course, we can have, let us say the reds are penicillin. So, they will remain like this in the water, while since it is insoluble, so biomass will come like this. Okay. So, basically biomass will settle. So, that is how you can separate, but can you accelerate this settling part? Yes, of course, you can accelerate this settling part by a process called as centrifugation. So, the biomass that is settling here, or the biomass that is over here, okay, and moves to here, the force that is putting on is mg, the gravity. Okay. And now in the centrifugation you replace this force by omega square r. Omega is your angular velocity, okay, while r is radius of centrifuge, okay, and then basically you can accelerate this settling. So, this is one property that we can actually use to separate biomass and penicillin. On the same lines, you can also use filtration. Does the filtration also uses the same property, the solubility and non-solubility? If you think about it, it looks like it does, but actually it doesn't. Filtration is based on size. Your biomass, so let me write it again here. So, okay, filtration, penicillin and biomass. Okay. What is the size of penicillin molecule? It would be in approximately nanometers. It is a very small molecule, right. What is the size of biomass? Biomass is typically in 1 micrometer, 1 to 1.2 or 1.5 micrometer. Okay. So, can we exploit this property to separate? Of course, if you see something like this, a filter, and these are solid material and these are the holes in the filter, okay, which is what filtration is. And the size of this hole, let us say if it is 0.45 micrometer, then if penicillin is going in and your biomass is going in, what will happen is biomass will not be able to come, it will stuck here, okay, while your penicillin will come out. Okay. So, that is how you can separate penicillin from biomass, A using filtration based on the size and centrifugation based on their settling uh, uh, settling capability. Okay. Settling is also indirectly linked to the size, the bigger is settles down, or let us say density, the more the denser settles down. So, there is a fine difference there. So, filtration is based on the size, while settling or the centrifugation is based on the density difference. Okay. Normally, we assume that bigger has a higher density, but that may or that is not normally, that is not always true. So, we cannot make it as a rule of thumb. Okay. So, remember this, filtration separation is based on size, while centrifugation or settling separation is based on the density. Okay. Now comes a little bit more challenging one. We have seen how we can separate penicillin. So, this one, if you look at the mouse, penicillin from biomass. Now, we have to see how we can separate penicillin from glucose. So, last time we used either size, size or density for separation of penicillin with biomass. Okay. But here we cannot really use it, because if you look at it, this molecules, so on your left hand side of the screen you see penicillin molecule, while on the right hand side it is a glucose molecule, and you can see that there is not really much difference between the size of these two molecules. I have just made the glucose cyclic, but it is a straight chain. Okay. So, if you break from here O, then it becomes 1, 2, 3, 4, 5, 6 carbon chain, and then you have an CO group. Okay. So, okay. So, as I was saying, penicillin and glucose, the we cannot use size because they are very similarly sized molecule, as well as we cannot use density because they are both carbon based. So, their density difference would not be much. So, now we have to think what are the other properties that we can use. Again, if you see, it is a cyclic structure, the penicillin, as well as glucose is cyclic structure, but glucose has lot more OH groups and it is highly soluble in water, while penicillin while changing the pH is not soluble in water, it is highly soluble in amyl acetate. Okay. So, what we can do is we can check solubility in amyl acetate. Okay. When you check solubility, this is high and this is for penicillin is high, while for glucose this is low. So, now we can use penicillin and now we can use amyl acetate to separate penicillin and glucose. Okay. So, what is a typical process looks like? Typical process looks like is this: you have a separatory funnel, okay. This is separatory funnel, this is your aqueous phase, and on the top here your amyl acetate. Keep in mind, amyl acetate is absolutely insoluble in water. So, they will always make these two phases. Okay. And what will happen is your penicillin molecules which are here. So, I will make them as cross. Okay. This penicillin molecule will move here. Okay. While your glucose, which is dots, they will remain here. So, what happens? Glucose remains in aqueous phase, while penicillin moves to amyl acetate phase. Okay. And then you can just recover this. Okay. So, you open this tap and next it will look like something like this, and only all this phase, while your it is here and it is separated, and then you can collect penicillin in amyl acetate. Okay. You can collect penicillin in amyl acetate, and basically that's then you can do further steps of separating it again from amyl acetate in phosphate and so on and so on. Whole of this process is essentially solvent extraction. Okay. So, there are other methods as well. So, here we are running it in batch, but you can also run it in continuous mode. You can also run it in a column kind of, and I will show it to you in couple of slides, next slides. Okay. So, this is how your these are what are the typical steps in the downstream processing. Okay. So, you do a fermentation, okay, and you harvest the broth, which is here, okay, with my mouse. This is here, and this is after your fermentation. The concentration of your product is 0.1 to 5 gram per liter, and the quality is 0.1 to 1.0% purity. So, here quality means purity. Now, you remove insolubles. What are your insolubles over here? The insolubles over here are biomass, okay, and either you can use microfiltration, okay, or centrifugation, okay, and the product quality still remains 0.1 to 5 gram per liter, okay. The concentration still remains 0.1 to 5 gram per liter, but the product quality improves slightly, 0.2 to from 0.1 to 1 it increases, it doubles, 0.2 to 2.0, okay. Here basically you remove biomass, and you remove biomass if your product is in broth, okay, and you collect biomass if your product is in biomass itself, intracellular, okay. So, there is an important distinction here. This is your bioreactor, and this is your cells, and this is your cells, and this is your broth. You collect the broth, okay. Now, in this broth, it's like this, okay. Now, let us look at the cell. If your cell is like this and your product is here, here, here inside the cell. So, you have an intracellular product. Recombinant proteins like insulin is a typical example of an intracellular product. If that is the case, what you collect after filtration or centrifugation, okay, after filtration, which is size dependent, or centrifugation, which is density dependent, you collect biomass, okay. You collect biomass, and what you discard? You discard your broth, okay. Otherwise, if your product is like this, your cell is like this, however, your product is like this, then again you do filtration and centrifugation, but you collect broth and you discard biomass, okay. So, keep in mind what kind of product we are looking for. If we are looking for recombinant protein, which is typically an intracellular product, then you collect the biomass. If you are looking for antibiotics, which is your extracellular product, or sidro4, which is your extracellular product, then you collect the broth and discard the biomass, okay. And this can be done by filtration or centrifugation. Filtration is size dependent, while centrifugation is your density dependent. Here we do not use nanofiltration, we use microfiltration, and this I will explain it in the next downstream processing steps that we are going to have a look at now, okay. So, now you have removed the insolubles, okay. You have microfiltration and centrifugation, and you have improved the quality by 2 times. The next step is isolation of product, okay. But before that, I must mention here, if your if this is your cell and this is your product, okay. Okay. This is your cell and this is your product, then you need to do a cell lysis. to basically bring the product in the solution. So, you do here before cell lysis you do a filtration or centrifugation. Okay. After that you do a cell lysis, which is this. Okay. What does that mean? That means you have broken the cell like this and your product is now outside the cell. Okay. And there are several methods to do this cell lysis. You can use chemical, okay, like NaOH or detergent or surfactants. Basically, they destroy the cell wall, which is this. Okay. You can use mechanical method like sonicator, which is lab scale, or homogenizer. Okay. So, mechanical method, sonicator, homogenizer. Sonicator is typically lab based, it is not used at the industrial scale, but homogenizer is used at an industrial scale where you put mechanical pressure on the cell wall and again break the cell wall. Okay. Then you can also use enzymatic method. Here basically these enzymes again eat the cell wall and destroy it. Okay. So, these are the methods that are used for cell lysis. Now, once your product is outside in the, you have broken the cell and brought it outside, then you can use, talk about isolation of the products. Okay. Here basically solvent extraction or adsorption, these are used, and basically you increase the concentration of your product from 0.1 to 5 gram per liter to 5 to 50 gram per liter, and the quality is increased by 5 more times compared to after removal of soluble, that is 10% purity. Okay. So, for isolation of product you have two methods, solvent extraction and adsorption. Okay. Solvent extraction is based on solubility, and the example we have seen here is of penicillin in amyl acetate. Adsorption is based on interaction. This interaction can be hydrophobic, can be complexation, can be ion exchange. I am not going to go into details of it, just wanted to tell you that this kind of interaction is possible. It can be electrostatic, okay. So, your interaction can be hydrophobic, complexation, ion exchange, electrostatic, or some others as well. Okay. And the example that we have seen is furfural adsorption on hydrophobic beads. Okay. So, these are two example, and keep in mind what I have shown you is that we can run them in continuous mode as well as we can run them in batch mode. Okay. Typically for adsorption you have columns that you can run, but you can also do it in batch like just a shake flask. Solvent extraction the same, you can put it in separatory funnel or you can do it in solvent extractor stage wise. Okay. So, this is just for your information. Now, the next step is purification. Here you can use precipitation, chromatography, electrophoresis, ultrafiltration. Okay. However, two techniques, chromatography and ultrafiltration, this is used largely and it can increase the concentration to 50 to 200 grams per liter, while the quality improves by 50 to 80%. Okay. If you remember, I have said in chromatography you always lose the you always make the make it dilute, which is true, but chromatography with solid phase extraction or chromatography with distillation or chromatography followed by precipitation, your basically the concentration would increase. If you just do simple chromatography, the concentration would always dilute. So, what exactly is chromatography? Chromatography is simply like this. Let us say you have a column, okay, and you have packed this column with your material. Okay. Now, what you have done is you have green is your product, while red is your impurities. Okay. You have injected this into the column. So, what it will look like is like this. Okay. Once you have injected, and you have an eluent that is flowing in. Okay. So, you always have some liquid that is continuously flowing in the column. After some time, okay, T is equal to, let us say T1, what will happen is this would remain here, okay, while your impurities might have gone till here. Okay. Now, if you do T is equal to T2, what will have happened is your impurities, your product is only till here, while your impurities have reached here, and at T is equal to T3, your impurities would come out, while at T is equal to T4, your product would come out. So, if you collect the fractions at time t is equal to T3, you will get the impurities, while collect fractions at time t is equal to T4, you will get the product, okay. And it will look like this. If you look at, if you can measure the concentration, and this is time, okay. So, you have injected and this is your time t is equal to T3, and then this is your time t is equal to T4. Okay. So, basically what is happening is when you have injected a mixture of your product and impurities into the column, where a continuous eluent or continuous eluent is flowing through, it can be 50% acetronitrile, 50% phosphate buffer at pH 3 or sulfuric acid at pH 2, things like this. Okay. What is happening is that because of the interaction with this solid, so we have solids, it is packed with beads. Okay. Because of the interaction of product as well as impurities, what is happening is that your impurities are not interacting with solid and thus passing through, while your product is interacting with the solid and thus slowing down. So, basically because of this, your the speed at which the product passes through the column is slower compared to the speed at which your impurity passes through. So, impurity passes through the column faster, while the product passes through the column slower, and hence there would be a separation. Okay. So, this is the principle of chromatography. Now, this interaction between solids and impurities and solids and product can be of different type. It can be electrostatic interaction, it can be hydrophobic interaction, it can be size dependent interaction. So, it is also possible that bigger sized molecule passes through quickly, while smaller sized molecule goes into the cavity of the beads and interact more, and hence passes through the column slowly, and this basically leads to size exclusion chromatography. So, that is one thing. Another step of the purification is ultrafiltration, or what we call as nanofiltration. So, basically in nanofiltration the size becomes even more smaller. So, here basically we talk about size in terms of molecular weight of protein. Okay. So, basically we talk about 1000 Dalton or 2000 Dalton. So, what we talk about is that nanofiltration with a cutoff value of 1000 Dalton. That means, so if 1000 Dalton is cut off of a nanofiltration membrane, what it means is that any molecule which is smaller than 1000 Dalton will pass through, while the molecule bigger than 1000 Dalton will not pass through. So, let us say this is a filter and the liquid is moving like this. The flow rate is this way, but the filtration is taking place orthogonally in perpendicular. What is happening here is that there is this small molecule and then there is this big molecule. The size of this is 500 Dalton and size of this is 1500 Dalton. Dalton is basically molecular weight of the proteins. Okay. It can be in kilodalton, typically 10,000 kilodalton or so. 500 Dalton is a small molecular weight molecule. If both of this molecule goes inside, what will happen is this would remain here, would remain here and comes out from here, and this is your called as retentate, while this is called as filtrate. Okay. And your molecule is coming here. Okay. What does it mean? It means is that it is same like ultrafiltration, where a bigger molecule is stopped while a smaller molecules goes through, okay, except the size of the pore has become even less. So, here instead of defining the size of the pore by nanometers or micrometers or in a unit of length, we are defining the size of pores by the cutoff value of the proteins molecular weight. So, 1000 Dalton, 2000 Dalton, 50,000 Dalton and so on and so forth. Okay. So, these are the two methods basically that are typically used for purification. After that, to increase the concentration, we can use precipitation. In chromatography, we cannot increase the concentration. In chromatography, we always dilute, but after that we can mix it, follow it with precipitation, where we actually can increase the concentration to a very large extent. So, for example, with glutamic acid at pH 3.2, it precipitates and you can increase the concentration by 10 times or so. Okay. Then the last step is polishing, which is crystallization. Basically there, as I explained earlier, you evaporate the water, you put the liquid at a critical stage where its solubility is exceeding, where the amount of liquid, that is amount of solid that is present, has increased beyond the solubility, and then you can do sometimes nucleation and basically leads to formation of the crystal. Normally you keep either decrease the temperature, you decrease the temperature, and since solubility is temperature dependent, normally the molecules are more soluble at higher temperature, product is more soluble at higher temperature and it is less soluble at lower temperatures, like desferoxamine E. It is a kind of a siderophore which is soluble in water at 50 degrees centigrade, but very less soluble at 4 degrees centigrade. So, if you decrease the temperature, basically you can decrease the solubility. Okay. And there, with crystallization is a very good purification step. You can increase the concentration. So, concentration can be increased, but purity can be drastically increased to 90 to 100% quality. Okay. So, these are the two examples that I would like to show it to you. One is the production of streptomycin from Streptomyces griseus. So, this example basically, if you know, this is your streptomycin, this is an antibiotic. So, as I said, basically this is your cells, okay, while your antibiotic is outside. So, what you do is we have to remove the insolubles. Okay. How do we remove the insolubles? We remove the insolubles by filtration or centrifugation. Now, we have and the this is biomass and this is discarded. Okay. Now, you have this. Now, you want to do an isolation. So, this step is isolation, and you do isolation by typically by solvent extraction. Okay. Then another step in between, if you have not achieved the quality, it can be chromatography. Okay. And after chromatography, you can have crystallization, right. Basically, you need to have an antibiotic and you need to do a formulation. So, you need to do a crystallization where it can be weighed quite equally, like let us say if you need 50% of the antibiotic and 50% of other ingredient for a formulation, then basically we need antibiotic which is easily can be easily weighed. So, basically we convert it into solid by crystallization. Also, it is easier to transport, it easier to weight, it is easier to put into capsules and so on. So, we do purification with crystallization. So, this is your isolation and this step is your purification, and then you do a formulation, and then that is the end of the story for antibiotic production. Another next example is of intracellular product recombinant insulin. So, here basically it is again you have cells, okay, and your product is inside. So, you remove insolubles and what you take is biomass. You collect biomass because your product is inside, while you have a broth and this you discard. Okay. Once that happens, you do cell lysis. Now, you have a protein that is there. Okay. This protein basically you might need to do a refolding of this protein because it might not be in the right shape. So, you can do a precipitation of protein to purify it, or you can do his tag based purification. Okay. So, I will not go into the detail what is his tag, but it is called as affinity chromatography. Okay. It is based on affinity chromatography where your protein X has a tail and it binds specifically strongly to a bead. So, it is kind of a lock and key kind of arrangement. So, that binds very strongly and specifically, and then basically you can separate X protein with his tag, while you have another protein let us say Y with no tag or no his tag. There are different kinds of tags, but let us say no tag. So, this will not get separated while this will get separated. Okay. So, this is how you will separate the protein, and then again you will go into the refolding, final purification if it is required. Size exclusion chromatography is also there to purify them, and then you have a pure protein. Okay. So, this is just two examples, but there can be multiple of steps involved, and you choose based on the product that you have and as well as the impurities that you have. For example, many times there are viruses, so you have to do an adsorption steps in between with activated charcoal and where virus can absorb, and you remove the viruses. If you have a lot of salt in it, then you have to introduce ion exchange where you actually get rid of sodium salts. So, there are several steps that are in between, but typically these are the main unit operations that are uh, that normally take place in the purification of a product from biotechnology or bioproduct that is produced from biotechnology. Okay. So, with this I would like to end this lecture on biosuppression, downstream processing, and in the next lecture we have to study the strain management and strain culture and all those kind of things, because now you have realized that how important is the strain itself, because if your strain can produce much higher quantities, so you need to make sure that you select the right strain. So, that is what how we do it, what is the importance of it. This is we do it in the next class. Thank you very much for your time, and I look forward to seeing you in the next class.